Hi there. This video is about fossils and rocks, rock strata specifically. So I'll talk about some of the types of fossils and then some things about a rock strata and how rocks and fossils can be dated. As I mentioned in a previous video, a lot of this started with Charles Lyell, who was a contemporary of Darwin. Darwin knew him and studied his Principles of Geology, which he produced in 3 volumes, about the time Darwin was going on his voyages. And so what Lyell said was that rocks are laid down in succession, and that the deeper you go, essentially, the older the rock is. Now, they can be folded, so that can be reversed, but in general, the deeper you go in the strata, the further back in time you can go. So essentially, you can do a little time travel, so to speak, by digging through rock layers. And this also implies that the organisms, the fossils that you find, are also laid down with those layers, so that the fossils that are in the deeper layers are organisms from earlier times, and the fossils that are closer to the surface are generally those that are more recent. So a lot of the layers around Utah and Arizona have been examined because a lot of times the land is bare and there are canyons, so that you can look through these different strata and determine how old they are and what fossils are found in those. And so here are 2, one from Glen Canyon and one just a couple hundred miles away at Arches National Park and the stratigraphy. And you can see that in general they line up, although the layers vary somewhat in their thickness. And at the top there are some differences as well, but you can follow these layers for hundreds and sometimes thousands of miles and connect them up into the geology for the particular areas. In doing so many studies have been put together, and you can find very specific types of layers during different periods in the past. So you can look at the Phanerozoic eon, and then within that is the Cenozoic, Mesozoic, Paleozoic, and those can be further subdivided based on the strata that are found in those different layers of rock. So you can see the Carboniferous region here, the strata, and that's characterized by a lot of plant life, so that they deposited, as they decayed, they deposited a lot of carbon, and you'll find fossil fuels being dug from these layers. And then you go into the Permian, and finally the Triassic, Jurassic, and Cretaceous, where you have dinosaurs within that area, and then moving all the way up to the Quaternary and the Holocene, which is what we're in currently. Among some of the deepest layers, you find the Archean rock formations, and finally, back even before that, the Hadean era, where the Earth was primarily molten at the surface and all the way through. Note that the timeline is not linear, so it's more or less logarithmic. So you can see the strata at the top, and these zones at the top are essentially narrower, but they don't show up this way on the graph. And the lower areas are sort of squeezed, so if you really put this on a linear scale, those times would be represented as much wider zones. As I said, these layers can be followed by hundreds and sometimes thousands of miles, and this is just what's called the Grand Staircase in Utah and Arizona. And so you can follow these strata and drive several hundred miles and go through hundreds of millions of years of evolution, starting from Cedar Breaks on the left-hand side. Bryce Canyon is there. It's a little lower. Zion Canyon, and then eventually over to the Grand Canyon, where you can see many of the earlier layers also. So if you start at Cedar Breaks, you'd be dealing with things that are in the hundreds of, about 100 million years old. And then by the time you get over to the Grand Canyon, you're back to the Cambrian era, about 500 million years ago. This is a diagram sort of squeezing all of that together in one diagram. So if you look from the top, that first line dividing some gray cliffs from pink cliffs, that's about at the 66 million year mark, 66 million years ago, when the dinosaurs went extinct. And then you can follow through to the Cretaceous, Jurassic, Triassic, and all of those you would find dinosaur bones within those because that's where when the dinosaurs were on Earth. And then you can go to the Grand Canyon down here and look at even earlier strata, all the way back into the Cambrian and the Precambrian. So that demarcation occurs at about 541 million years ago. Here is a photo of Cedar Breaks in Utah. So this is the upper portion of those layers in the Grand Staircase. So you can see pink cliffs at the top, gray cliffs and white cliffs, and then vermilion or red cliffs down below, representing a period from about 200 to 50 million years ago. And there's a line there that I've drawn that you can see between the pink and gray cliffs, which is the KT boundary, the Cretaceous Tertiary boundary, at 66 million years ago. And that's where the dinosaurs went extinct. So you would find the dinosaur bones below that line, but then absolutely no dinosaur bones above that line. And that if you sample that layer, it's high in iridium, which indicates extraterrestrial, that is a meteor source for that iridium. And in each one of these regions, Cedar Breaks and then Bryce Canyon, Zion Canyon, and the Grand Canyon, because the rivers through those areas have dug down into the strata, you can, from the top to the bottom of each one of those canyons, examine different times from the past. So by the time you work your way down into the Grand Canyon, you're examining older and older layers from about 250 million years old to nearly 600 million year old strata. And you can actually see these layers so that you can determine by color or by consistency, etc., which layer you're in different parts of the Grand Canyon, as well as other canyons in that Grand Staircase area. So here, some of those layers in the Grand Canyon have been labeled so that you can go back in time into those different regions, going from the top, which is about 250 million years ago, down to this lower one shown here, which is closer to 500 million years old. This is a cross-section through the Grand Canyon, going from the top there, which is somewhere around 200 million years ago, to the Precambrian, which is greater than 600 million years ago. And so you can work your way through those different layers and find fossils representing those time areas or those time periods. And then you see down in between the Cambrian and Precambrian, there's a major shift in the crustal formations at that time, and then a reorientation of the sediments that were added after that time. And right there at that transition zone, once you enter the Cambrian, then you get that explosion, that increase in animal diversity that is unprecedented at any other time on Earth. Another thing that can be done with following these strata through different parts of the world is it has also proven the plate tectonics theory, that is, that the different plates around the world have moved over time, so that you can connect different rock formations from South America to Africa, and from Africa through Madagascar, India, and Antarctica, and then through Australia as well. And not only are the rock strata continuous in those regions, but the types of fossils that you find also are continuous throughout those regions. Additionally, you can look at the wearing on the rocks and specifically see where glaciers have moved, because they scrape along the bottom and leave linear marks on those rocks. And you can put together the continents this way also, based on knowing where ice caps occurred on Earth, and connect them up and bring them back together to where they were at the time the ice was on each one of those areas. Many of those things can't really tell you the antiquity of each one of those layers, but you can determine the antiquity of certain layers based on measuring the decay of certain radioactive isotopes of elements. So uranium-238 decays and emits a helium nucleus called an alpha particle, and then has a half-life of 4.5 billion years. So half of the remaining uranium is there after 4.5 billion years, and the other half has been converted to thorium, a daughter nucleus or daughter element. So by measuring the amount of uranium-238 in a rock compared to the amount of thorium in that rock, you can date it as according to making a graph of the half-life of uranium-238. By using a variety of radioactive isotopes, you can date things of different ages, so that uranium-238 and 235 are down at the bottom, and that has the half-lives of 4.5 billion and 710 million years old. So you can date things in the range of about 10 million to 4.6 billion years ago. Rubidium-87 can be used for 10 million to 4.5 billion time ranges. Potassium-40 can be used for younger ages, and then carbon-14 can be used for even younger ages. And so by choosing the appropriate one, you can find a range where you can date particular rocks fairly well. So this shows that process. So when you have uranium in a rock, when it crystallizes, then in one half-life, it might be a billion years old, and then two half-lives, it's 2 billion years later, and in three half-lives, you'll see it's less of the uranium is around and more of the lead in this case, and it's 3 billion years old. So you can date them according to how much the proportion of uranium and the daughter ion or daughter atom, in this case lead. You can then graph those according to the amount of the parent atoms versus the number of daughter atoms and the number of half-lives, and then come up with a scale for each of those isotopes. So in the case of uranium, you could go back to about 4.5 to 4.6 billion years on this time scale. For carbon-14, then the 1, 2, 3, 4 half-lives would be different, so that out at 5 or 6 you'd have 30,000 years versus 4.5 billion years for uranium. So scientists have used a variety of methods for dating strata, dating fossils, etc., going back all the way to uranium, thorium, potassium, argon, amino acid racemization. This is where amino acids convert between the L and D amino acids. Radiocarbon dating, that is carbon-14. Tree rings you can use, and you can actually look at the age of lichens also to get a dating for particular, within particular time scales. And then there's fission tract dating, and I'll talk about that in a few minutes. One method for dating fairly recent fossils is to use carbon-14, nitrogen-14 ratios, because when you have a decay of carbon-14, there's a beta decay and it becomes nitrogen-14. So at the time the organism is living, it's incorporating primarily carbon-14 into its biological molecules. But when it dies, it no longer is adding carbon-14, so the only thing that's occurring is changing that carbon-14 into nitrogen-14, and there's a certain rate of decay, a certain half-life of that, which is used then to date how old that fossil is, up to sometimes up to 100,000 years, which is fairly recent in terms of geology and evolution. So essentially, when you're using radioactive decay measurements, you assume that there's a certain starting point when either the rock was crystallized or the animal was alive. And then over time, certain radioactive molecules in there will decay. And since no new ones are being added, then you can look over time and compare the parent molecule with the amount of the daughter atoms that are in there and determine the age of that specimen based on that radioactive decay proportion. In fission tract dating analysis, the rock is polished, a crystal surface is polished, and then you allow the decay to occur. The decay of the radioactive ions or atoms releases beta particles, which are helium atoms. Those actually are pretty damaging, and so those leave tracks in the crystal, and you can look at those microscopically, either before you irradiate the surface or after you irradiate the surface, and you can detect then the amount of radioactivity in that particular sample, and then determine what the original amount of radioactivity was, and now how many of those atoms exist in that sample. So those types of analyses are more difficult, but they also lead to dating things that are in the 1 to 4 to 5 billion year old range. So here's just one micrograph of one of those crystals, and you can see the tracks left over from the helium ions that have been released, the beta particles that have been released through radioactive decay of a particular element. So the fossils and the rocks surrounding them can be dated using radiometric methods, but now I want to talk about the fossils and how they're used to study evolution. So fossils can be of many different types, so that here you have remnants and impressions of certain animals, trilobites, fish, dinosaurs, human skull that's actually not completely mineralized, so it really is still mainly bone. And then you can have footprints, you can have worm tracks, you can have tunnels dug by certain animals, and a lot of those things will tell you a lot about the organisms that lived at that time. So the rocks will tell you something about the fossils, but the fossils tell you something about the rock strata as well. So at certain points in time, trilobites and other organisms were very numerous. And then there was a mass extinction about 251 to 252 million years ago. And so in the strata above that, you don't find any trilobites or these other organisms that essentially disappeared in a mass extinction. You can also look at a fossil and determine the approximate age based on other fossils that have been found in that same stratum, even from a different continent. So the fossils themselves can tell you something about the time when those existed, and the rocks can also tell you about the timing of those events, so they can be correlated. In particular, there are certain fossils, usually some of the more numerous fossil species, that are found characteristically in each one of those strata, those rock strata. So often those are some of the first things found, and that those will pretty much date that rock stratum without having to do radiometric analyses in every case. And not only hard-shelled organisms such as seashells and snails and things like that, but also the bones of animals can be used to identify particular times in history in certain strata. So in the Cambrian, it's mainly fish evolution that's going on, and by the time you get up to the more modern eras, then, or time periods, then you have more mammalian fossils. And of course, fossils of hominins and hominids, including Homo sapiens, Neanderthals, Homo habilis, etc. Certain taxa can also be used to identify major extinction events, so that before about 250 to 252 million years ago, there were a lot of different species of trilobites, and so they predominated. But then after that time, no trilobites are found as fossils, so they went extinct. And also from about 250 to 66 million years ago, belemnites and ammonites predominated in the fossil beds. But then after about 66 million years ago, no more of those were found from 66 million years ago to the present. So there was another major extinction event at 66 million years ago, which also killed all of the dinosaur species as well, and about 70% of all other species. The fossil evidence also tells us a great deal about how different groups of organisms evolve and speciate into other species. So one example here is of the glyptodont, which is an ancient relative of the modern armadillo, and you can actually pick out different times of these glyptodonts and look at the evolution of various body parts in those organisms. In Australia also you have a wombat ancestor and then the wombat itself, and you can also go back millions of years to look at the changes in that lineage as well. Entire phylogenetic trees also can be produced to show these various stages in evolution. For instance, the evolution of an ancestor, one branch became the hippopotamus and related species, and the other lineage led to dolphins and whales. And you can look at the dentition, that is the teeth, the tails, the limbs, etc., changing over time in these different lineages of both the dolphins and the whales and all of the ancestors that came before them. Watching this transition from a land-based, a terrestrial-based organism to an aquatic mammal that has lost all of its major limbs that have changed from legs into fins, or in some cases have disappeared completely. And also the transition from a tail that is primarily for balance and other purposes into a tail that becomes a fin and a means of propulsion in aquatic and marine environments. The bones can be studied also to examine the evolution of those structures. So 50 million years ago, there was an organism, an ancestor of whales, that had legs and feet and a tail. And again, the tail is partly for balance, and a femur and pelvis that were articulated so that the animal could walk on land. About 38 million years ago, that organism, one of the descendants, started living primarily in the water. There was a change in the structure of the femur and the pelvis, such that it didn't need legs to propel itself on land, but instead it had vestiges of legs at that time. And eventually descendants along those trees, those lineages, developed into much reduced structures, primarily from mutation, so they weren't being used, and so any mutation really wouldn't hurt their function. And so there's a reduced pelvis, and there's still a femur and part of a lower leg, but certainly no foot structure or anything like that. And there's no external part to those bones, so they don't form any function in propelling that organism at all. So this is one way that this fossil evidence supports evolution. For example, if the whale doesn't need a pelvis and a femur, why would it even have one? It has no legs in the rear. But those are vestiges left over from when its ancestors did walk on land, and then transitioned eventually into aquatic and marine environments. And then those means of propulsion, those legs and feet were no longer necessary. So they pretty much mutated out of existence. In the 1800s, a very unique fossil was found in Germany, and it was looked like a dinosaur, but it had wings, and so you could see the feather imprints in the fossil. And so this was the first time anyone ever found a fossil of a dinosaur or dinosaur relative that had wings. And so there's a transition then. This is a transitory organism that was still a dinosaur, but it was then evolving along the lines of where the modern birds are. It was named Archaeopteryx, and it was about the size of a modern-day crow. So it could fly, but probably it was a gliding bird rather than a flying bird. And here's what it looked like. It was a small bird-like organism with teeth still. So it didn't have just a beak, but it had teeth. And it was a transitory between dinosaurs and birds. And more recent molecular studies have shown that modern birds, if you look into their genome, still have the genes for producing teeth rather than just beaks. But they've mutated, so they no longer form teeth now, other than an egg tooth, which is used by the chicks to crack their way out of the egg when they're hatching. Also, more recent studies have shown that many dinosaurs did have feathers, although the initial feathers they had were rudimentary and were probably just for heat retention and not for flying. Later, other types of feathers developed or evolved, and so there were gliding birds. And then finally, there were birds that had feathers for powered flight, and that's the modern birds. And so Archaeopteryx could glide, but it wasn't really good at flying, a powered flight for hopping into the air and flying. I would like to point out that there are many other types of fossils. So the shell in the middle and the shell down at the lower left, those are ones that have been substituted by minerals. So those are the typical fossils that people think about. But there are also many shark's tooth fossils. Sharks shed their teeth. So one of the most frequently found fossils are shark's teeth. Also, there are dried specimens. The one at the top is just a dried mushroom. The seeds in the middle, those are from pack rat middens. Pack rats gather seeds and plant material, bring it back to their nest, and they actually urinate on it. And the urine, when it crystallizes, becomes hygroscopic. That means it will bring water to itself, and it dehydrates other things such as the seeds and the plant tissues, and it actually preserves them for tens of thousands and even hundreds of thousands of years. And plant sap, that is, plant resins, entrap both plants and insects and other things, up to small birds actually, which then solidify into amber. And so some amber is up to hundreds of millions of years old, and it has entrapped insects and plant tissues and preserved them completely. It excludes all oxygen, so there's no degradation of the tissue. So you can look at fine details of organisms in amber. And you can even extract DNA sometimes from those samples up to hundreds of millions of years old. And these tubes on the right, there's some tissue in one of them, and the others are liquid. Those are either dried specimens that have been collected, or it's actually DNA that's been extracted from ancient materials. The thing on the upper right is a piece of cedar wood from King Midas's tomb. He was a Phrygian king that lived about 4,000 years ago, and then they built a tomb which consists of cedar wood on the inside and then some spruce on the outside, and then mounds of dirt. And so this thing is about 100 feet tall, a hill, and inside is his tomb, where his body was kept, was buried. We were able to extract DNA from a piece of that tissue, and discovered that even though it's supposedly Lebanon cedar, it matches Lebanon cedar to an extent, but it's quite different from the modern Lebanon cedar. And so we think that the cedar that was used for his tomb was related to Lebanon cedar, but it is actually a species that's now extinct, that no longer exists. Oh, I should mention that his tomb, if you ever want to go there, is in Gordion, Turkey. So it's modern-day Turkey now. Just like animal fossils have been used to look at the evolution of animals and other types of organisms, plant fossils are quite prevalent around the world, and so you can see that during certain times ferns and tree ferns predominated, and then it was only more recently that gymnosperms such as pines and cedars and things like that evolved, and then later angiosperms, the flowering plants, evolved from there. Here's just a really rudimentary timeline of plant evolution. The first unicellular plants that then diverged into glycophytes and red algae appeared about 1.9 billion years ago. But then by the Cambrian and later, there was a more diversification in plants. Both green algae appeared, and then so there's a whole line of red and green algae from 1.9 billion to the present. And then bryophytes, or mosses, and then ferns, which predominated from about 410 million years ago up until about 100 million years ago. And ferns were one of the first rudimentary vascular plants. About 360 million years ago, then the conifers or gymnosperms appeared, and one of the offshoots were the ginkgos at about 250 to 300 million years ago, and then the cycads, some of which still exist today, also in that same time period. And finally, the flowering plants appeared about 130 to 140 million years ago. And almost all of the data on this came from fossils. More recent molecular work has solidified these dates and also added to our knowledge of the diversity of the evolution of the plants. Here's a little more data on that, based both on fossil leaves, but also fossil seeds and fossil spores, etc. So you have about an expansion from the progymnosperms and the ancient plants about 417 million years ago, as an expansion from those unicellular red algae and green algae. This is primarily from the green algal line. And then you have both the gymnosperms that expanded and diversified, and some of those have gone extinct, as you can see by the ends of some of those groups. And then evolution of the angiosperms, the monocots and dicots. But more recently, we've split those up. Our taxonomists and molecular taxonomists have split those into monocots, eudicots, and magnoliids, that's groups that are like magnolias, and some other groups also. Again, most of this information has come from fossils, both from fossil leaves, seeds, spores, etc., and more recently from molecular data has solidified a lot of these taxonomic groups and evolutionary patterns within plants. So ages on Earth were determined by stratigraphy, that is, looking for natural layers in the rock that represented each one of these different periods of time. And generally, as you go deeper in the strata, you go back in time, although, as I said, you can have folding of those strata so that in some cases they're actually upside down if the rock has been folded completely over. The ages of the rock and the fossils have been determined by radiometric methods, but you don't have to use these methods for every single fossil hunt or identification. What is done now is you look for specific fossils in the collection of fossils at where you happen to be digging. And there are specific fossils that appear only in certain strata, and so that can be used to date the sample as well. So it's become a little bit easier now to identify particular strata and the age of fossils, dependent on what other fossils are in that stratum, as well as some occasional radiometric testing to assure that you're in the right time period for dating of that particular fossil. Okay, so that's the end of this video. Hopefully you enjoyed it, and I will talk to you later. Thanks.